So today we'll be discussing a rat model of human gene expression for aggressiveness and its significance regarding natural selection and self-domestication. The specific paper we will be looking at is a rat model of human behavior provides evidence of natural selection against underexpression of aggressiveness-related genes in humans. And this paper was a collaboration between researchers from the Institute of Psychology and Genetics in Novosibirsk, Russia, and the Natural Science Department of Novosibirsk State University, also in Novosibirsk, Russia. This paper is a part of the final or third part in a series of papers regarding the natural selection against the underexpression of aggressiveness, through which they studied different rat lines that were analogous to human hereditary lines. And also they looked at self-domestication syndrome. Now, my interest in this basically comes from an epigenetic view. Of course, the connection might not seem obvious. I mean, what does epigenetics have to do with the natural selection against underexpression of aggressiveness? Well, I mean, it might be that epigenetic changes alter the expression of aggressiveness-related genes. And through that, we might see a change in the selection for different genetic or genotypes. So what's in store for us? Well, we're going to take a look at the introduction, which is the basic overview of behavior, natural selection, gene expression, and qPCR. We'll take a look at the hypotheses that the study goes along with. We'll look at the experiments set up in pre-qPCR procedures, the results, and the take-home message. The introduction. What's up? Well, what is behavior? There are two primary kinds of behavior. There's learned behavior and innate behavior. Learned behavior is behavior that's gained through experience, where innate behavior is behavior that's encoded genotypically and is brought about through the reaction to very specific stimuli. Now, this innate behavior is dependent upon gene expressions that are dominant or subordinate, where subordinate gene expressions are expressions that are typically docile, where dominant expressions are more volatile or violent, aggressive, so on and so forth. Now, the aggression aspect is focused on in the study, as stated by the title, and the importance is that aggression results in the dominance or social hierarchies that are evident in all known species of animals. These social hierarchies are typically shown to be favored by natural selection because they introduce a higher quality of life and greater longevity of life for individuals of the species. Factors that influence gene expression, you have pleiotropic and epistatic effects. Pleiotropic effects are where one gene expression alters the gene expression of two or more other genes, and epistatic effects are where one gene expression is dependent upon the gene expression of another gene. These are sort of the inverse of each other, not exactly, but very close to being the inverse. And these are controlled for in the study by selecting specific genes from the string database that aren't known to have pleiotropic or epistatic relationships. Another factor that can influence gene expression is epigenetics. Epigenetics are, epigenes are basically stress markers on genetic material that alter the phenotype without altering the genotype. Those are harder to control for, but in this case, if the epigenetic expression and natural selection are working in the same direction, then they sort of cancel each other out. So possible flaws in the assumption about aggression in this study. This study assumes that natural selection, it states that natural selection favors aggression, but assumes aggression is solely positive, where aggression is not necessarily positive. For instance, greater aggression can lead to conspecific violence. In this case, in this image to the right, you see two chimpanzees that are that are in conflict with one another, right? And the issue with conspecific violence is that it can actually work against, don't get me wrong, it's still an aspect of natural selection, but it can work against the optimal selection, meaning that though there might be one dominant animal organism that has that is dominant because of these aggressive traits, the other one that's subordinate and gets into the violence with the dominant one might have more traits that are more survivable, that are better for survivorship in the environment, but because of his subordinateship, he might have less opportunities for mating, which means less opportunities for passing down that beneficial genotyping. The secondary aspect, of course, are strained relationships, which come with social hierarchies or dominance hierarchies. And these two things, violent strained relationships, among other factors, can lead to or are aspects of hierarchical instability. And hierarchical instability means that there can be turnover rate in the hierarchy, and this turnover can actually lead to physiological changes that might cause organisms to basically be able to mate less or produce less beneficial offspring, even if they have a beneficial genotyping. And this hierarchical instability is somewhat dependent upon the verticality of the hierarchy. The more vertical it is, the more unstable it is. So they need to know. Well, you need to know what natural selection is. And natural selection is, as a summation, a basic summary, is the process by which the survivorship of individuals determines the species genotyping as a general. And then there's sexual selection, which is considered an aspect of natural selection, where an individual of a specific sex of a species desires certain characteristics from an individual from the other sex of the species. But then there's artificial selection. Artificial selection is where certain traits are bred for either to maintain or enhance them in an organism. In the image to the right, you can see mustard, and its artificial selection for specific traits that it has results in a variety of crops we use today. Domestication is the process by which subordinate traits are bred into an animal, generally by human beings. And outbreeding is the inverse of inbreeding, where inbreeding is the mating of the most closely related individuals in a species. Outbreeding is the mating of highly differentiated individuals of a species. So the study looks at two areas of the brain in particular. The hypothalamus, which on the right is the gray object near the center of the image labeled hypo. The hypothalamus is the fear controller. It also controls defensive behavior. It's an intermediary between the nervous system and the endocrine system, and it works as an intermediary using the pituitary gland to release hormones. And then the periaqueductal gray, which is the orange component in the image that's labeled PAG. The periaqueductal gray is also controls defensive behavior, but the majority of the defensive behavior controlled by it is maternal defensive behavior. And because the subjects of this study are male gray rats, it is acting as a control. So the gene expressions in question are the following. All of these are aggressiveness related genes. CACNA2D3 is a protein encoder for the calcium channel complex, which mediates the influx of calcium ions into cell membrane. Deficiency of this causes iron anemia, which in turn causes anxiety disorders. Gene knockout of the GAD2 causes sensitized pain. Sensitized pain just means that you're more sensitive to pain, including just basic touch. The SYN1 is the underexpression of SYN1 results in over-anxiety. And SYN1 specifically is known to be exaggerated through epigenetic means. qPCR, basic rundown. Quantitative polymerase chain reaction is a method of measurement of gene expression through the amplification of genetic material. The main things you should know is that it consists of cycles in which the genetic material is grows from exponential to linear and then plateaus. And also that the main measurement here is the threshold, which is where exponential changes into linear. So the hypotheses. Well, first we have to ask the question. Does natural selection favor the expression of aggressive or domesticated subordinate associated genes in humans? The complications to this are the aforementioned pleiotropy and epistasis, or the interdependence of gene expression. Also, the ethical consideration for animal experimentation. It's difficult to use animals in experiments, especially when those animals must be executed in order to conduct the experiment. Though in this experiment, they follow the EU guidelines for animal experimentation. And then, of course, there's also the studies trying to focus generally on humans. Due to the things that will be mentioned further, you are unable to conduct these studies on humans. So the hypothesis, the specific hypothesis is that there will exist significant overexpression of the aggressiveness-related genes in the hypothalamus of the aggressive rat over domesticated rats, but that the significance won't exist in the periaqueductal gray. The general hypothesis is that natural selection acts against the underexpression of aggressiveness-related genes in humans. So the experiment. Well, the approach to experiment is somewhat simple. They used an outbreeding of rats gathered from a previous study along the same line. This outbreeding, they outbred two strains of rats. In one, they artificially selected for aggressive phenotyping, and in the other, they artificially selected for domesticated or subordinate phenotyping. Of these, they took 6 aggressive and 6 subordinate, all of them being 4 months and all of them being of similar weight class, also all of them being from unrelated litters. They took them, they decapitated them, they removed the hypothalamus and the periaqueductal gray from each of them. They flash froze them in liquid and nitrogen for preservation, stored them at -70°C. And then they extracted the RNA using a reagent. They quantified it, verified its integrity, and they synthesized complementary DNA using a primer and thermal cycler. And then they conducted qPCR using the RQL30 as the gene reference. This basic process can be seen on the image to the right. And then finally, they analyzed the data using the Kolmogorov-Smirnov test, the Fisher Z test, and the Mann-Whitney U test. So the results, what they find? Well, initially, they found an unexpected non-normal distribution that invalidated their Kolmogorov-Smirnov test results. But as the figure on the right shows, which is based on the Mann-Whitney U test, there were significant differences between the two. Now, the bar graph, the white bars in the bar graph, they represent the domesticated or subordinate rat line, and the gray bars indicate the aggressive or dominant rat line. The bar height indicates the mean expression, and the error bars indicate the standard error of the mean. The bar graph on the top, the A series, is the gene expression differences in the hypothalamus, where the B series, or the bar graph at the bottom, is the differences in expression in the periaqueductal gray. The asterisks indicate significance, with 2 asterisks being more significant than a single asterisk. As can be seen from the hypothalamus series, or series A, there exists a significant difference between the expression of these aggressiveness-related genes amongst the subordinate and dominant rat through all gene categories, whereas in the hypothalamus, the periaqueductal gray, series B, this does not exist. There is no significant difference for any gene expression. So, what are the findings? Significant overexpression of aggressiveness-related genes in the hypothalamus of aggressive rats over domestic rats. Also, there is no significant difference in the expression of aggressiveness-related genes in the periaqueductal gray of aggressive rats over domestic rats, which was expected, hence its use as a control. The conclusion, There exists significant overexpression of aggressiveness-related genes in the hypothalamus of aggressive rats over domestic rats, but this significance does not exist in the periaqueductal gray. Using the string database, which is a database of protein-protein interaction, this would suggest that the natural selection acts against the underexpression of aggressiveness-related genes in humans. What is the significance of this? Well, this means that natural selection favors greater expression of aggressiveness-related genes in the hypothalamus over underexpression, but not in the periaqueductal gray. This does not mean that overexpression is greater than underexpression. For instance, there is an aggressiveness-related gene, COMP, whose overexpression actually reduces aggressiveness. Also, another aspect of significance is that this could lead to better model organisms for human disorder analyses. For instance, in this specific study, it found that these gray rats might be a model organism for iron anemia in humans because their iron anemia is a consequence of a homologous gene structure or protein structure encoded by a homologous gene. And this means that it might be a better model organism for iron anemia than, let's say, another organism that also undergoes iron anemia, but through different genetic means. Take-home message. So let's look at the bigger picture before we get to the final take-home message. Epigene-induced gene expression may impact natural selection and result in less aggressive offspring genotype, thus resulting in wasted genotype. This is complicated, but basically what I mean is that epigenes may cause they may influence gene expression, they may influence phenotype, giving the false expression that the genotype is a certain way, which means that those of a specific genotype that is more beneficial might not have many offspring. Secondarily, our current trait distribution may be a result of natural selection favoring aggressiveness, which suggests that early humans may have been more peaceable. Though, alternatively, kind of contrarily, this also leads to the idea of self-domestication. Now, the reason why this is contrary is because self-domestication is the idea that humans have become less aggressive over time. And you might ask, well, how does this data suggest we've become more aggressive, that natural selection favors aggressiveness? Well, the specific thing here is that there are two main kinds of aggression: there's proactive aggression and reactive aggression. Whether or not these gene expressions lead to proactive or reactive aggression is to be seen. But either way, if they lead to proactive, then it doesn't necessarily indicate that it's supportive of the self-domestication theory. But if it leads to reactive, then it's definitely in contrast to the self-domestication theory. So the self-domestication theory was a theory brought up by social Darwinists that was recently repurposed and reintroduced through evidence. It's the idea, well, domestication itself is the idea domesticated traits are shared across species, but not with their wild type ancestors, which is interesting. And these traits, of course, are cranial size, brain size, the roundness of the cranium, also the flatness of the projection of the face, tooth size. All of these are shared, as well as a more complex sexual selection and also longer mating period. These are all shared among domesticated animals. But what's interesting is the idea of self-domestication. And self-domestication, we've long known that cats, for instance, self-domesticated themselves. They did it twice. And what that means is usually an animal becomes selective in order to take advantage of a commensalist way of life. For instance, humans produce waste, rodents attract to the waste. Cats take advantage of the fact that rodents are attracted to the waste produced by humans, right? And so in order to do that, they become less reactively aggressive than they once were, because they don't want to react to humans aggressively. They don't want to end up harming the thing that provides them food, right? Of course, it's not a conscious decision, but you get the gist of it. Well, cats for a long time, cats were the primary focus for self-domestication. There weren't many other animals considered self-domesticated, but recently we've become to think that dogs, because we've considered dogs domesticated, but we've come to think that maybe dogs were actually originally self-domesticated, because it makes sense, right? How does a human go about domesticating a dog, or really domesticating a wolf into a dog? There's extreme risk involved, and the likelihood that you can get two to mate, even if you take them as puppies, you'd have to get multiple puppies of different sexes. It's now, while it's possible, it's not highly plausible. So the idea is that wolves originally self-domesticated, and then humans domesticated them further. Well, the self-domestication theory applies to a further extent, whereas it suggests that humans also went through this process where we self-domesticated ourselves because we share these common domestication traits. So the take-home message, well, natural selection significantly favors aggressiveness-related gene expression in human hypothalamus as examined through a rat model. Natural selection does not significantly favor aggressiveness-related gene expression in the human periaqueductal gray, and underexpression and overexpression of aggressiveness-related genes can lead to disorders. And that's basically the gist of it. And these are my citations. So thank you for watching this. I hope you learned something here today. I tried to do a good job presenting it. Thanks for your time.